guest speaker. This is Mrs. Winnie Sloan here. She is part of the ABE-LA CSUCI program, but she has been with the ABE-LA program for over 30 years. So she knows her stuff. She's going to share an implementation PowerPoint with you. So without any ado, take it away. Thank you, Winnie.
All right, well, welcome. So my name is Winnie Sloan. If you have been with the program for a while, you might know me as Winnie Litton. I was that for until a couple of years ago when I got remarried. And so any documents or shared documents that say Winnie Litton on them, you have access to anything that says that. You don't need to ever ask my permission. And everything here that I'm sharing with you today, you can copy it, paste it, change it, whatever you want to do. How many of you have ever used Pear Deck before? Look at that. And how many of you started using Pear Deck because of distance learning? Yeah, that was me too. So I had I still have a smart board. Does anybody have a smart board? Okay, there we go. So, and Promethean, because I used to in-service in that, I didn't work for the company, just in my school district. And with the software, I guess is what you would say, is called Notebook, that was in the Notebook presentations, and it just wasn't as accessible for the students to get a copy of it. And so I used to do a lot of printing as PDFs and then sharing those to students. And when distance learning came about, I just took the bite and just made all my presentations go from Notebook to Google Slides, and it was really the best decision I made because it's so accessible by students. And if you don't know, Pear Deck is just a layer on top of your Google Slides, and it allows for interactivity. So I encourage you to join this, and the reason why is once you join this, I won't test you, I'm not putting you on the spot or anything, but you could. But I won't. But when you join, then you will have every single resource, because what I'm sharing with you, you'll have access to, and then any link that's on there, you'll have access to that. When this presentation ends and I end the session, it'll show up in your Google Drive. You'll also get an email, and you'll get a Google Doc that goes along with it. And it is really, really helpful for kids. Especially because if kids are at home, and I think we all know we're going to see more and more of kids feeling free to stay home instead of come to school. It's part of distance learning, and students are able to join the Pear Deck from home. I've had kids come into my classroom from hospital beds, from airports, because of the way I run my class, and I'm happy to share any of those strategies with you. I am an open book. You can ask me any question and I will answer. So right now I can see 11 students have connected, and so I'm counting 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. So we have 10 children that are still waiting. Can you check to see if your neighbor knows how to get onto this presentation and offer assistance if they need that? Say, may I help you? Are you struggling? So if you haven't yet, all you need to do is go onto any browser, and then you're going to type into your browser joinpd.com. That's the first thing you want to do on any browser. And then once you're in there, then it's going to ask you to put in the code for this presentation. You type in, they put them in 3-letter chunks. You do not need to put a space in here. They just do that because for your brain it's easier to process in chunks of 3. So it's COSOQA. Now we have 16 students. How many of us are in here today? What did I count?
I can do it.
23. Okay, thank you. All right. Again, check on the left and check on the right and see if they're in, and say, may I help you?
You just got to do this part. Okay. Yeah, you did.
Now, what I'm modeling for you right now is what I would do in my classroom. Yes.
Have you had it kick you out once you choose your account and send it back to Google? Because that
I
happened to
have
me
not.
did it to her as well, on our phones.
Oh, on your phones? No, I have not experienced that before. Yeah, try your school account maybe.
Yeah, I use my phone and I use my school account.
Yeah, use your school account.
I used the same account both times. The first time it picked me.
I am
and
so
the second
sorry.
time it picked me, hers was
It's
kicking
okay, it's okay, I'm okay.
You're okay. She's all, don't draw attention to me.
This is my buddy right
You're
here.
throwing me under the bus here. So, I have a little bit of assistance here because if in my classroom, what I would be doing is I would have two windows and I would drag one window to my smartboard and I would leave one window on my desktop and then I would change the presentation as I went through there. And what it allows me to do is if I had embedded questions in here, this really wasn't a Pear Deck in service, it's just that I'm showing you what I use so you can see on the resources that I have set up. There's a lot of formative assessment that's embedded in there to check along the way because you and I both know, if you've been teaching, if all you did was student teach, you and I both know that you'll go, does everybody understand and what does everybody do? Yeah. So they don't understand and we have to be really careful, at least for me, my number one passion, which I could start crying about in a nanosecond if you ask me to, is science literacy. I think now more than ever, people need to have those critical thinking skills that they need to make good decisions. And we have seen the outcome of poor decisions again and again and again. So I guess you could turn, I don't know which volume that is, it's probably me. What I really am passionate about is being able to teach my students those critical thinking skills and enable them to make those choices. Now, the Amgen Lab, like they said, I've been doing this for 1 year minus the inception of the program. When it started, Hugh Nelson was, thank you, Hugh Nelson started this program. He was a teacher at Newbury Park High School. Who has heard of Hugh Nelson? Okay, so amazing man. He worked at Newbury Park High School. He did a summer teacher in-service. Amgen used to have teachers do in-services and learn technology over the summer. He did that and wanted to bring the lab with a man that worked at Amgen. I think he was a VP, Bruce Wallace. Do I have this right, my Amgen expertise? Yeah. And Bruce has since passed away in a hang gliding accident, if I recall. But when they wanted to bring this into the classroom, so he brought it into to Newbury Park, and then the next year Oak Park High School got involved in the lab. And so there were just our two high schools, and it's a crack up to me that it's an international organization now in the sense of presenting of the lab. When I used to go pick up my supplies, I kid you not, I would drive up to building, I don't remember what it was, building, I wish I could remember, but it was one of the early buildings at Amgen. I would park literally in front of the building. Every time I picked up supplies, I would bring Hugh a banana. That was because I knew he liked bananas, and he seemed to be hungry for healthy food, so I would bring him banana. We would walk in the doors, literally like this, no lock. We open the doors. I would go into the lab prep rooms and pull things off of shelf. Oh, I need some Gloria broth. Oh, I need some Petri dishes. This is how it went. There were no gates, no nothing when we first started that lab, and we would pick up those supplies. And so the lab has really evolved over these 30 plus years. But what I'm eternally grateful for is that my students got to experience it every single year. And you have seen, if you have been participating authentically in these labs as you progress through this week, it's hard work, right? Are you envisioning kind of a sense of overwhelmedness in how you're going to manage this? There is no sense of overwhelming that I have not experienced personally myself that you could ask me about. And the strategies I'm trying to give to you today are ways to alleviate and help and learn from my mistakes. There is literally no mistake you could make in this lab that I haven't made for all of my students and got to reap the rewards of that. What I've been grateful for is whoever your contact person is, you call them, You explain what's going on, and they will help you to the best of their ability. But you know, just like your virginity, there's some bells you cannot unring. And I tell this to my students. I'm a biology teacher. So there are some mistakes you are going to make that I will not be able to fix. And so we have to keep working around that. I hope that doesn't now make it so you can't present it. But if you've ever seen it. So I'll go ahead and get started. Anything that is a shadow is a link. So if you see here, that is a shadow. That is a link to my school's website, my website on there. And I share things out quite a bit. Are any of you teaching AP Biology? So I share quite a bit on the AP Bio Facebook page. There's nothing I won't give you. Anything that helps students be scientifically literate, I am on board. And so you can find resources there. Every lecture I've ever given is on my YouTube channel. And the links to the notes are on the YouTube channel as well. All my notes are there. So that is a good jumping off point if you want things, my YouTube page. And then are some of you aware of TikTok? So early on in the TikTok when it started, I was like, you know, like you have a group of kids that you can smell the trouble on them. Do you know what I'm talking about? They're bless their little hearts. You know, they're just trying to navigate high school, you know. And so I always try to connect with them. Like that's how I read the Twilight series books. Is, you know, that's how because there's this quiet girl wouldn't talk. She's reading the book. And I'm like, oh, what are you reading? And she goes, do you want to borrow it after me? And I'm like, sure. You know. And so the kids knew I had an Instagram and a Facebook page, but Facebook page is only if you're like over 50 now, which I am, 55. So that's so out of date. And so there was Instagram and there's whatever. But anyway, this group of girls is like, and you need to have TikTok. And I'm like, TikTok? I'm like, okay. So we made a video together. The first video, the camera, I'm not even holding it the right way. The first video is like sideways, and I think it has 16,000 or something views. I have no idea now. But it just kind of took off. And there weren't a lot of biology teachers on it initially. Now there are quite a few. And I have a little over 100,000 followers. And I don't do anything that is, there's nothing, all my things are just quick science videos and encouragement and study habits. That's the limit of what I'll do. I'm not into getting viewers. I'm not trying to get more. I don't make any money off of it. I just try to, again, science literacy is my passion. And if that helps some kids, then I'm all for it, and it doesn't hurt me to try to make a quick video. So I incorporated a few of those as well. Okay, you are so perfect. So this is what I would say. You did the thing. You signed up here, and you're here this week. You're doing it. You're learning all the steps. You're like, how am I going to do that every time? How am I going to do this with all my kids? So these would be my big 3 tips. Number 1, get their brains ready to understand the material. Conceptually, they need to understand why they are doing something. If they understand why, they're more likely to be able to apply that to any new situation, right? So you really have to build that academic foundation. This is not a discovery lab opportunity, okay? This is not where like, hey, let's see what happens when we mix an acid with the water, discover it, okay? If you're gonna maximize this, you need to lay a firm foundation. Number 2, get your classroom ready. This is not going to surprise you. You know when you're going to do it. You have it scheduled. For instance, I started prepping for next year's lab during this year's lab. As soon as I finished it, I began prepping it. So I have TAs that are in my classroom, students. Now in the past, COVID put a stop to all of this. I had nobody that had any previous experience pipetting, and they will tell you in the Amgen materials that you can have kids pipette for you everything ahead of time, and so they have to be really good. I mean, they really have to be good and trained in the lab for you to trust them to pipette anything other than water, you know, but they can get all your water tubes ready. Don't let them pipette enzymes unless you know that this kid is like, you know, awesome. But I at least had some kids who could help and had experienced that, but then we went through COVID, I had 2 years of no ABE-LA, so I didn't have any kids with that experience. But you know what they can do? They can label microfuge tubes for you. So if you're doing this lab, let's say you're doing it in November of this school year, And if you have assistance from any student, you bring out your microfuge tubes, they will give you a bag of microfuge tubes. Yes, won't you? Okay, so you can have those already labeled ahead of time and racked up per each lab. Those are things you can do to get ready ahead of time. I do a lot of prep. However, you're going to present to the students whatever presentation over the course of the lab, have the entire presentations over the course of the lab done before you start the lab, right? Does that make sense? Like what you're going to talk about every single day, however it is you present, make sure you know what you're doing each day because you're going to keep going back to that. And number 3, you need to provide a really good, authentic curation process for them to record the lab, however that is. And I'll give you a couple different options on that, and obviously share with you my latest evolution of what I do to have the students curate. Also, you need to journal down things that you learn about yourself as you do the lab, and tell yourself, don't forget, set the water bath the night before, all those comments that you need to. I don't know if they give you a lab notebook anymore with all the teacher instructions, or you print it out. However you're going to do that, make notes to yourself about each lab so that you remind yourself ahead of time what you need to do. Okay, so disclaimer, I know many of these strategies I'm preaching to the choir, you already teach this way anyway. I'm not making an assumption that you are not all magnificent teachers. And I'm just trying to give you one strategy that I hopefully it's useful to you. Take with it, manipulate it however you want. All right, perfect, let's keep going. So number 1, be sure that students have the foundational knowledge to understand and appreciate the lab minimally. This would be my list. I'm sure Amgen has a list. Remember, you don't need to write all this down. It's in this presentation. And even if you're not in here, you will get a copy of it. They will make sure that you get a copy of this presentation. But I would have taught prokaryotic and eukaryotic cells, DNA structure and replication, transcription, translation. I would talk about control of gene expression at whatever level, okay, that you are teaching. And then I would already have had a whole discussion on biotechnology and what's out there right now. And when I am teaching those, because prokaryotic and eukaryotic cells, for me at least, is early on in the year, when I am teaching those, I am always talking about the Amgen lab. The Amgen lab is woven through all my discussions, not formally, but informally. Bless you.
Thank you.
So when I talk about DNA being a code book, and once they understand how the DNA nucleic acid gets into the protein language, I tell them, you're going to be doing this later on in the year. You're going to manipulate a code book, and you're going to take a eukaryotic gene, and you're going to put it into a prokaryotic cell, and you're going to get that prokaryotic cell to make whatever you want it to make. So I'm talking about it, anything that has to do with the lab, I'm talking about it through the year. So by the time we get to the lab, they are like ready because I have been promoting it the entire school year. So that is one thing. It gives them a purpose for learning them. When you guys are far enough in the lab where you've done lab 1, tools of the trade, lean heavy into that lab. Now, in the past, I have made students cry. They were so scared during gel electrophoresis that their hand was shaking, and they're like, and I go through the gel, and they're like standing to the side. I'm like, what are you doing? And I'm like, there's no crying in biology, and reference movie clip. But I do stress upon them to take it seriously, and so I said, today you're moving dyes around, and you have microfuge tubes, and they're huge, and they have so much in them, and it's all colored fluids, and you're going to be sucking up some small amount, and you'll see the different colors. I go, guess what? Next lab, when we do it, everything looks like water, and you have so little of it that you might want to come up to me and say, I don't think there's anything in my tube. If I could count how many times people say, I don't think there's anything in my tube. I'm like, yeah, there is, And so when I explain to them about centrifuge, when you talk to them, you've talked about pumping, right, with the micropipette. I have them pumping every single time. You can pump, I've done it, it doesn't matter. I have them pumped so that it becomes second nature, so that they're always mixing those solutions in there. And so lean heavy into tools of the trade. Lean heavy into that you're like, you're going to be doing this at this point in the lab. You're going to be doing it at this point in the lab. So wait, could you go back and then go to this? I think, here's a little TikTok for you explaining an overview.
This is a real B&H customer story. Jack and Barbara, professional wildlife photographers and B&H customers.
Here's a little analogy to help you understand the flow of information in a cell. I want you to think of your DNA as like a cookbook of information on how to make, let's say, pie. How do you get that information out? Well, you open up your cookbook and you write out a recipe card. That would be your mRNA. Then you take that recipe card to your kitchen counter, that would be a ribosome. You start putting your ingredients together, those would be amino acids, in order to build your protein. What CRISPR technology can do is it can actually go back into your recipe book in your cupboard and start altering the instructions inside the cookbook itself. What viruses can do is they can insert their own recipe cards into your cell directly, so you start making their protein products. In fact, what some viruses can do, retroviruses can do, is they can take their recipe cards and turn them into pages in your cookbook. Hope that helps.
So that is just a little quick video that I would use. This presentation right here.
This tutorial will teach you how to use BLAST. BLAST is a bioinformatics tool used by...
But I have a video on how to do the BLAST lab, if that is helpful. Okay, so I'm not going to teach you a lesson on everything in here, so don't stress out. What you will see right here, just because whenever I talk, I always like to tell, like, you know, kind of this is how the flavor of my classroom is. I have, off the slide. Okay, so what I try to do is, how many of you are familiar with SEL? Okay, so I got a deep dive on that because when COVID first started and they were sort of stressing out and the kids stressing out, a counselor said she would help me and we would be putting an in-service together for all the teachers in my district on SEL. So I said, okay, sure, yeah, I'll do it. So I didn't know anything about SEL, social emotional learning. I know that much now. The counselor couldn't help. So I just was researching, and we all did those things we needed to do, right, during distance learning, but I did this deep dive research on that and the importance of making sure you value the kid more than you value your curriculum. And I tell my kids, and I get all cry again, I tell my kids I love them every day. Every single day. Now I'll tell you what I can't do is remember their damn names. But it doesn't mean that I don't care. Sorry about the swear. It doesn't mean that I don't care about them. I know all their stories. That's just a problem. Every year, #lifetime goals is I try to learn all their names. This year might be the year that it happens. I'm really hoping, but I tell them I love them every day. I remind them that whatever's going on in their life and all their other classes, I don't assume that I'm their only teacher. I don't assume that I'm their only one that gives them homework, right? I look at the whole child every single day. Now, will the whole child make stupid, poor choices? Yes, they will. And so you just very, matter of fact, say, boy, that was a poor decision. So here are the consequences. What did you think was going to happen when you made that decision? Okay, and move on, right? And it's okay. Or we're really working at teaching them when they fail, right? That's when you learn, right? You've heard it and you all know. The mistakes I made, I'll give you one mistake right now that I made. We used to have to make the cells be competent. Now, you lucky ducks, you get competent cells delivered to you in a little box, and your biggest problem is keeping that thing cold enough so that they remain competent, right? We used to, back in the day, I can't believe I'm so old that I can say this, back in the day, we had to walk 10 miles to the lab and then make them competent. So you have to soak them in this, tell me if I'm wrong because I don't know the strategy, calcium chloride, spin them down, and calcium chloride, spin them out. Then you're supposed to store them in the calcium chloride solution and then put them in the freezer. You know what I did? This is what I did for all 6 sections that I was teaching. The last one I put in there, luria broth. Okay. So I just spent, a couple days growing bacteria, spinning. I just made them not competent. not competent in any way, shape, or form. Little did I know that I had made that mistake. All 6 of my sections, I don't have a prep period. They pay me to teach an extra period, so I'm always stressed out. And so all my classes, the only growth they got, if you can guess, any guesses? Tell your neighbor, what plate do you think I got growth? Are they far enough?
They haven't done that yet, no.
Oh, yeah, the control group, the Luria Bra plate was the only one. Zero transformation. Here I am getting them ready for the lab. We're in, day 5 of the lab, and I screwed it up for all of them. As I tell you, there is not a single mistake you can make that I have not already made for you. Now, do you think I ever made that mistake again? No. In fact, when I realized what I had done, it's like I got sweats. I was like, what did I do? I screwed up, And what I did is I told the kids, I screwed up. I'm really sorry, but you still learned all those skills. I'm sorry we didn't get the evidence of it. If I could take it back, I would. Can't unring that bell, which they've heard me say. And then we move forward on that learning opportunity. And that's what we need. We need to model making mistakes, acknowledging those mistakes, and how we would do it better. So on this would be, this right here, what I was really after to tell you is that I ask these kind of questions every day at the start of class. And let me tell you why you want to do it. Selfishly, if you care about your kids learning your information, talk about them as a whole kid first before you start in on your information. Because I guarantee your information is not as important to them as whatever else they're carrying around in their emotional backpack, which is much, much heavier than the one that has their books in it, right? So if you want to be effective, that's why I always lead with these types of questions. So the next slide, if you go to that next slide here, this would, I put calendars up and what we're doing. My homework, and this will be important as we get into some other aspects of this, when I give my homework, it is prep work. It is preparing for the next day's discussion so that I can move more quickly through the information. I don't give drills or terms or any of those things as homework points. I make a lot of resources available to my students. What they choose to take advantage of, hashtag poor choice or hashtag good choice, that's on them. The only homework I have is prep for what we're going to be talking about in class. So when I give a lecture, a discussion on a topic, they have already watched an ed puzzle on it. They've already taken notes on it. My students take what's called group shared notes. They get in collaborative teams of 4, and they're all contributing to the notes. And that way, if you're absent or you're at home, and you can go on my website and find more about that if that is something of interest, or at the end when we have questions, you can ask me about it. So they have already learned the big ticket items or have that as a resource. So when I'm in class, it's kind of like a flipped strategy. I can just get going on whatever that topic is. So you'll see, if you just kind of go one at a time, just kind of click through them. Yeah, you can go fast on these. This is like go back one. This is a really good one. Have you seen this mouse? I think they have a monkey now too. I might have updated it. Talking about, nobody cares about a green mouse, right? Nobody cares about a green mouse, but that's evidence that you got a gene into every one of those cells, into a mammal cells, right? So that's significant. And that's something I think you want to reiterate with the lab. I know they give you a storyline about insulin, right, in the lab a little bit. I don't have them do all the work behind that, but I do talk about it. I said, you're going to be changing DNA. It's not like you can look at the cell, if it was making insulin and go, you good little cell, look at that insulin you're making. You have no evidence of that, and I tell them that's why they're making cells that have antibiotic resistance, and this is why they're making cells that glow red now. It used to be glow green. That's why you're making that. But you could have done that with another gene as well. So this presentation is yours along with other ones. Yeah, now we can just kind of click through. You can see how we talk about cloning. This one right here, this is why I like to do Pear Deck. Highlight what you are most interested in biotechnology. And there are some really good videos. I don't know if you have seen that glowfish. If you click on that one. You don't have to watch the video. Here it is, yeah. But this is like a commercial for glowfish. That's a great catcher, on the kids to get their attention about that. And then you can keep going. And there are some videos. This is the video that's from, you have it from the ABE website as a resource that talks about
Using genetic
one.
engineering.
This is, you can play that if they, so they can.
Cells that produce proteins they wouldn't ordinarily produce. For example, circular pieces of DNA, called plasmids, found inside bacterial cells, can be engineered to make such proteins as human insulin by inserting the gene that codes for that protein. Special enzymes called restriction enzymes, which recognize, bind to, and cut specific DNA sequences, are used to open up a plasmid in preparation for the insertion of a gene. Some restriction enzymes leave the DNA with uneven ends, also known as sticky ends, shown here. If the gene we wish to insert and the plasmid are both cut with the same enzyme, these sticky ends will be complementary, meaning they will readily bond with one another. In a follow-up reaction, gene fragments, cut plasmids, and an enzyme that connects DNA fragments are mixed together. This so-called ligase enzyme attaches the sticky ends in the solution to produce circular plasmids that now carry the gene that codes for the desired protein. Note that the efficiency of recombinant techniques is not 100% and may lead to plasmids with no inserts or multiple inserts.
So when I show that video, when I'm talking about biotechnology, I start talking about the Amgen lab, right? That's a really good introduction to it. So it's not the first like an onion, you keep layering it and keep approaching it from a different way. So I'll incorporate it in there. If you go, can you scroll back up? There was another one. This right here. Oh, sorry, it's there. So I didn't used to talk about phosphodiester bonds. There's not like any expectations from NGSS about phospho, but it helps then when you're talking about the Amgen lab, when they're reading about phosphodiester bonds, it makes more sense, and this is a great place to incorporate it. So you will see that. You will see down here on slide 18, there are other where it says stay tuned. I have other videos about restriction enzymes. So even though this particular presentation happens months before the Amgen lab, for AP and I think right before for honors when I do it with them, I am already alluding to all of the parts of the Amgen lab at that point. Okay, so we can keep scrolling through. Now, and let's go to, here, perfect. And then we'll go to the next slide. Okay, so then my next one is prepare early, okay? Don't we tell this to our kids? I was working with some new teachers this year and it drove me absolutely nuts because they were lesson planners the night before and they said that's how they work best. It's not how you work best, okay? All it does is you're modeling for students, wait till the last minute, then you're working under the stress hormone, you're working under adrenaline, and you're saying that that is the only thing, a chemical push to get you to get your work done, right? And then we know the effects, the long-term effects of stress. So for me, as a teacher who has taught for 34 years, I think I'm going on, 35, anyway, I ideally, and I still change. Like I can remember my boys are grown and out of the house, but I remember they'd be like, Mom, haven't you taught that like 20,000 times? Why are you working on that lesson? I'm like, because it can be better, you know? And I update and change every single year. I am never done, much to the regret of my new husband. And he updates too, but he's one of those people who does it in the morning, like at 7 o'clock or something. That stresses me out. And so I like to, and it's for me a better life, if I know if I'm going to update any lessons or presentations, it's all done the week before I'm ever going to use it. I talk about the kids about photocopying. I don't do a lot of photocopying anymore. It's Google Drive and Google Doc. It's sustainability. It's what I work on. But you know those teachers, right? Who it's like the day they need something, and then they want to cut you in line because whatever they're doing is more important just because, you know, and they've got to get in there, and then the photocopy machine breaks, and that's terrible, right? So I would recommend that you get all your ducks in a row as early as you can in your presentations. So when I say label and Alcott and store tubes, like I mentioned before, all my tubes are already labeled for April when I do this lab next year. They are sitting in a little box. Day 1, digestion day, ligation day, everything is labeled and good to go. So that's not giving me anxiety, right? Because once I get the solutions, whatever I can pipette early, I will. I already have the tubes ready to go, right? And when the kids are working on the labs or debriefing, I can be pipetting right then and getting ready for a lab that's, you know, coming up the next day or the next day or two. So get that organized. Whatever your presentation is, this right here is my most current iteration of my presentation, which you will have. Oh, here, we'll watch a funny first. We'll watch that. This is me getting ready. So that lab bench is where they pick up their daily supplies. That is their stock box. And so I put whatever solutions they need in that stock box for that day. Their everyday materials are at their lab stations. Right there, see the box? Okay, so that just stays there. And the whole reason I made that TikTok is because I wanted Thermo Fisher to give me one of those. I am sad to report they didn't give me squat. I thought, look, I'm showing Thermo Fisher, you know? But no, they did not. So this, this right here, this is a freebie that I got. You know, they give those, you know, they give, Karen would do it at her presentations, like you could go and get stuff. So these are like big racks, and so I would set up all the tubes, and they're lined up. I'm fortunate enough to have a refrigerator in my room, so they're all lined up and stored, ready to go. So I'm not freaking out about it. Okay, now we can go here to this presentation, and if you're in here, this should be a link for you. Again, you're going to see some SEL, you know, questions. I always lead. It doesn't matter how busy the day is. I always check in with them first. So you're seeing a lot of that. And then if we go down to this slide right here. So these buttons right here, I start with this because it's at the beginning, but these are linked to different pages in the presentation. So when I'm on day 3, I hit this button and it jumps to that, because this whole presentation is like 80 slides long or something like that. And so you can go, and if you don't do Google Slides and you do PowerPoints, you can screenshot any of these things, or you can print any Google slide. You can print it out. If you're still using Notebook, you can print your slides. You can't print your interactivity, but you can capture the images that you have on your slides in order to make these. So the very first one, this one right here. Or you can hit the one on there, it'll go there. Learning how to use the micropipetter, and also these, I tend to put there so it can take them to the home page if they want to at any point during the slideshow. So this is demonstrating how to use the micropipetter, but as I told you, they've already watched an Edpuzzle. They've already prepped for this, so this isn't the first time they're seeing it. So let's go ahead and just click through.
Question?
Yes?
I don't know, the peer deck isn't advancing forward so we can get to that.
Oh. Yeah, and one more. Oh, one back.
I think
That, there, yeah. So if we leave that, they'll have access to it, and then we can go back into that presentation. Perfect.
Thank you.
Okay. So on here, let's see if we go to this page. Let's see, slide 8. You're so awesome, Liz. I tell you that. I appreciate you. So I'm talking to them about how to use the micropipetter, but I'm also teaching them that day how to use the microcentrifuge so that they can practice with that. And I kind of leave some, now their guiding lab document that we use, I'm sharing that with you too, so just keep in mind, this is what I'm presenting. They have a different document where they're recording and has their directions on it, and I'm going to show you that one as well. Okay, so I'm just trying to keep it straight here for you. This is what I am presenting. So I am reviewing how to use the micropipetter, the microcentrifuge, and you can just kind of click to the next slide. These are screenshots out of directions that I'm giving them or out of the lab book. I have them, and I don't know if you have mini ones to use. I do. Mine are like early evolutions of mini one. This is a link, which you don't need to show them. It's like 5 minutes long. This is an explanation to teachers on how to set up their mini one. I have them watch it. You know why? Because when they do the lab, do they need me? No. Because they watch the teacher explanation, for teachers on how to set it up and the boxes and the solution, so they know how to do it. And they get to practice it once during Lab 1, Tools of the Trade, so when they do it on gel electrophoresis day, they know everything that they need to do. The only thing, remember how you saw those little stock boxes I have at the station for them to pick up and return every single day? All I do is I set their gels. I have their gels already made. I don't have them make it. I already have the gels made, and they're sitting there and they just have to pick up the gel from their lab station. That's how I handle it. And so, let's see, these are just more instructions. You can just kind of, if I were you, I'd just like click through the slides. Okay, go back one now, sorry. So when you're done, this is what I go through before I release them. I'm telling them their expectations. At the end, your table needs to look exactly how you found it, and I encourage them to take a picture of it. I do this for every lab. You should take a picture of what I have at your table, because that's how I want it to look. Because I don't have time to clean up after them afterwards, and I tell them, if I am, you're going to see it come out of your lab grade, and it's just going to be because I'm annoyed. Okay, so it's just going to be you annoyed me points. And because that meant I didn't get to eat something, or I was rushed when I went to the bathroom, so whatever, So life is a series of choices, make good ones. I tell them what solutions they're going to pick up and what they're going to return. I don't let them throw away anything unless it's the transformation day. Have you talked about transformation yet? Okay, so later when you're working with the bacteria, they're putting that kind of stuff in contaminant trash in those tubes, but I make sure, even though each class gets their own, each group, each class gets their own tube PDR, I don't let them throw it away, because those little white boxes that you saw there, they return them. What I do is take the lids off of all of them. I take a Ziploc bag that says like day 1 on it, and I throw all those tubes in day 1. And those may end up going in the trash, but right now I hang on to them for all my sections, because if I do have a kid that screws up, I'll go, oh, you need more PBR because you screwed up? Well, dig through here and try to find 3 PBR, and you'll have to spin them all down and try to suck them out. It's a life lesson, right? I'm not going to do that for them. They can figure out how to do it, but at least I have some resources for them to use. Okay? So I tell them what I want. Now, you'll see right here is a Google form I have made, and I am sharing with you in a folder that's linked to this, a Google form debrief for every single lab. Okay? Because I have a hyperdoc that they curate, and I'll give you some other ways you can do it too, but my current iteration is they have a hyperdoc of the whole lab that they record. I ask questions. They work collaboratively on it as a team. What a hyperdoc is, if you don't know, it's just a Google Doc with links and pictures. Okay? And it is a curricular pathway for them to follow. So they record everything into that hyperdoc, and there's a link on that hyperdoc to a Google form, which they work on collaboratively as the team. Now, you have all been in lab groups where one person does all the work, and then you have what I call hitchhikers, right? They're along for the ride but don't pay for the gas, which is apparently very expensive right now. And I said, you need to work collaboratively on that, and if you don't understand an answer to the question, you're going to be held accountable for it on your quiz or your assessment, so I would be contributing. Also on their hyperdoc, they have to own up every single class period what they contributed to the team that day. They have to type their name, and they have to say what they did to contribute. And I said, if your teammates don't agree with you, they're going to say, not at all. Did not do this. okay? And I go, if you're not working well, I go, you're not entitled to work with anyone. I only have enough for 8 stations, but if your group just says, I can't work with you, then you get Das Boot. And then you go and try to find some other group that will take you on, because they don't have to keep working with you. It's unfair. Now, that gets established way early on in the school year. Nobody wants to get kicked out of their lab team. You know, and I'll tell them a story. It's usually lab team 4. It's everybody who's gotten kicked out of every other team, you know, and they work back there together. They're the slowest. Their work's not any good. So if you're hoping to be in that team, don't do anything, you know. They don't want that, you know, and so they do kind of their own self-policing. Okay? So that's all part of the Google form. Then the start of the actual lab, let's see, do you know? Right there, yeah, either one, okay, perfect. Welcome to today. So this is the digestion lab, and then we'll go to the next one. So I have them talk. Do you know how to use a micropipette? Do you know how to use a centrifuge? Do you know how to use a gel electrophoresis? They can type up, like, what would you tell somebody how to do that? And in Pear Deck, now this one we haven't started a Pear Deck on, so you can't answer it here. You can only like do one Pear Deck at a time, right? So I'm just showing you this, but if I was running this with kids, students write your response. Now you'll see some other slides where instead of saying students write your response, I've embedded a multiple choice question. Or it says students can draw, and that way students take notes on all my slides while I'm talking. They can use their stylus with their Chromebook, and then they have that as a Google Doc later, and they can use that. So that's what that is. So I go through what it means to be antibiotic resistance. You can see the 2 different plasmid rings over here. If you go to digestion, we talk about, they've already had to answer questions about what those genes are and what they represent, and why we're trying to move this RFP silent gene. There's no way over here from pecan, there's no way to express it, so we're going to cut it out with restriction enzymes. This is a good time for you to talk about why there's 2 restriction enzymes instead of 1. When we first did this lab, there was just 1, but you know, if it has the same recognition sequence on either side, it could go in either way, and that decreased the possibility of getting, you know, the transformation. So now we have 2 different restriction enzymes. It's a really good discussion. We talk about where they cut at. Let's see, let's go. Yeah, perfect. We talk about size. I tell them size matters. And we talk about what we could predict later about that, since they've already learned about gel. So I'm always alluding to the next step and the importance of this step and what happens if they screw that step up. And so they take it very seriously. Here, you can see, you can either use a water bath, which is here, or this wonderful thing from Thermo Fisher, who they refuse to give me anything. Which, if you get an option, like water bath or this, if they give you an option, choose this every single time. Okay, it's so much better. Okay, and then your supplies, if you can go to your supplies. All right, so you're gonna pick up from the table 4 clean microfuge tubes. So that's somebody's job to go get those tubes. In your white stock box, you have these 4 solutions. So I don't like the way that's written, but hey, I had a TA do it, and I like it better than me having to label all of those tubes, so I'm willing to live with it. Okay? I then also put a colored dot. You can see the dots here, you can see the dots here, so they know what they're going to get, right? I've already shown them a picture of what they're going to get, so that they're not stressed out about it, and they can go through it. Keep in mind, they have access to this presentation, so they can go to this slide and go, okay, did we get everything? Now, one thing I do is I have some colored tubes. I wish I had more. I need to order them this year. But this one right here is water. It is different from their practice water. I give them a new water, and it's in a different color, so that I know it's nice, clean, distilled water, and they don't get it confused with other tubes, and I keep that one pretty full. I also use this opportunity because I'm on a smart board. Can you see down here, the like meniscus, I guess is what you would see right here? And I say, it's down there, and this, it says right here, students draw anywhere on this slide. They can literally use their stylus and try to find where the solution is. That solves so many problems to when they say, I don't think there's anything in here, because you can see it and they draw it on here. And I go, look at the RE, I didn't give you very much. And it's down there. You need to make sure that your tip is in that solution. Okay? If you don't see it in the tip, it's not there. The other thing we always use is never dip without a fresh tip, hashtag life lessons, okay? And so they will say that, you know, and they, you know, about holding it up vertically, all those things you're teaching them on day one. That's why I say lean into tools of the trade, because you're gonna think forward thinking to every single lab where they're gonna need to know how to do that. You've gotta see it in that tip. Okay, all right, let's keep going. What is the very next slide after this? That's the one I wanna show them. Okay, this is also a very helpful slide, in my opinion, okay? Because this right here is the steps of what they need to do. And so on here, it's telling them put buffer into all, you know, that's what you're gonna lead with, right? Then in step 4B, you're gonna put some pecan into K plus and K minus. But over here on the left, I put a column of how much I provided them with. So I'm telling them, I gave you 20 microliters, how much are you going to consume? Well, 4 times 4 is 16. How much should be left then? Just 4 microliters. So you have enough, right? That helps them in their planning, you know, because they'll be like, you didn't give me, yeah, I did, you know, I could have screwed up, but I bet I didn't, you know, and, but that really helps so they know how much they're starting with in each one of these solutions. And this gives you a chance to talk about control, you know, control tubes and their importance. So then, let's see, if you go to one more, when you are done, then I tell them what it should look like, what they should respond. I'm prepping for the next lab, which is you need to label your lig tube and bring it to me, because what I do is I have them label it on that day and turn it up front to me, and then I pipette in the ligase at the bottom of each of their tubes, so it's already there and they pick that up from me the next class period. Okay, and then we can just kind of click through. So I talked to them about the importance, why we need to denature those scissors. Here on, yeah, you can click on this one. Yeah, perfect. Yeah. We talk about what should be in the tubes, what I've already given them in those tubes. And you can go through, you know, on your own time and kind of look through each one of these steps. A couple things I would point out if we keep going, 43. Right here. I have them draw what are some of the different combinations they could get, make sure they draw the one that we want. Right here, 46. Again, I have pictures. This is what's in your stock box. You already labeled the lig tube, so when they're looking at the instructions, I say we've already done this, we've already done this. Here's what you're going to do each day. We can jump down to gel electrophoresis. Perfect. Read in my mind. What's the importance of gel electrophoresis? To verify we cut the plasmids, that we cut them in the right place, and that we ligated them. Gel electrophoresis day, let's see, let's go to 52. That's a big day. Have you done gel electrophoresis yet?
We're doing that next.
Oh, fun for you. Okay. So you're taking samples out of all of your tubes, just little samples. You've got to leave it behind. You know, you just leave, you know, you're taking, 4 or 5 microliters, I can't remember, out. And then you're checking to see if you have cut and if you have ligated. And so what I do is I make their gel tubes be a totally different color, whatever color I have, so they don't accidentally pipette out the rest of their experiment and then they don't have anything for the next step. So I literally, before they load their gels, I have them return all their stock, you know, their original tubes to me so that they only have yellow tubes at their station. I teach a little process of like which order to go, which will be, you haven't done that yet, so you can look at it later. But one on 54 that I find really helpful for kids, especially if they have 4 different solutions that they need to load. Whatever you're loading into, you make it empty and put it right on the edge of your microfuge, your block. And then whatever needs to go in it, you put it behind. And then as soon as you have loaded out of that into it, you close it and it goes back. And you make it hop back one row. And that way you know you put that solution in. You will see when you do gel electrophoresis that that's one of the problems that they have, is they can't remember what they loaded. And because there's no color change or anything, and it's hard to change, verify with volume, that you want to teach them a strategy. Once it gets loaded, it gets moved to the third row back. If it hasn't put in whatever you're putting in, put it right there at the front. So there are different pictures here on gel electrophoresis. You can go all the way down. Oh, here, right here. I have them predict what they think their gel will look like. I show them a gel that I did. If we keep going on transformation day, I break it into sections. The prepping of the tubes, getting them ready, chilling, then I go on to talk, kind of break up the day for them on their transformation day because it's a lot and they can only hold so much information. And so slide 62, I say, okay, do all of this and then we're going to talk again. And I show them pictures of what it should look like for them. And then if we go to slide 64, I try to say, first you do this, second you do this, you're going to be piping in here, which once you've done the lab, that'll make more sense to you. We can go to 65. And so it talks about the importance of heat shock. Again, lots of steps. Go to 67. I talk about when you get to your plates, how to label plates. There's another one here. And what Amgen does is, I talk to them, there's so many plates that they make, they cannot put labels on everything. They used to. They actually used to stick them on the bottom. But what you need to do is pay attention to the lid and the base, and the only marking you have to know what's in that plate is on the lid. So if you change your lids around, like if you take them off and then put it back on like a little clamshell game, you won't know what's on that plate. So all of these have those kind of strategies or things that I have learned over the years to talk about, go over with them before they actually do it. Okay, and this talks about taping. I also have charts in here about predicting what the results are. And if you keep going a little more, I have images of plates and what they actually look like and what they should expect. Okay, so that's what I use to present. Okay, now I know I've given you a lot, but I only have this short time with you, and so I know I'm inundating, and I'm hopefully, are you doing okay? Okay. All right, stand up one time and tell somebody next to you what's one thing you can use so far, and then I'm going to finish up with another document I want to show you. Go ahead, stand up. What's one thing you know you can use? Stand up, it's good for your belly. Down. Let's
You can show me a million pictures.
Okay. So, on this last part about curation and reflection, it is a twofold for me. One is a hyperdoc that they work on as they're going through, which I tell them I can look at any point whenever I want to, and I can start grading it. So always be current on it. Do I? No. I just grade it at the end because I'm busy and I don't want to. So, but I tell them I will, and the threat sometimes is all that you need. And then what gets graded every single day is that they have I actually don't grade, I grade it after a few days, I really, but they have, it's due. So I have a Google form which they work on collaboratively as a team, and that is asking content-based questions. And so those Google forms, and in fact we can click on that one right there, I think it's a link. those Google Forms, if you scroll down, what are the names of your group members, alphabetical order? Yeah, I'm that OCD. It's easier for me to grade if they are in alphabetical order. They have to capitalize their last names, their first name's just the first letter, what class period, what station are you working at, and then if they did the reading, in the introduction of this lab, pre-reading, it was explained that what you are doing in this lab is similar to the techniques and processes to make what? To help those suffering from a common problem. So I ask them questions. Now, this is a Google form, so what you really want is you want it from my Google Drive, which I have for you. So all you have to do is, then you can edit them for you. You don't have to remake them, just make a copy of them, okay, because we're in a Google form right now, so you don't want to edit from there. You need your own version of it. So I have a link to a Google Drive set up just for you. You can go in there, make a copy of them, change the name, change whatever you want to do. It's right there. That's a link to it. Okay, so I have one for every single lab. Yes?
Do you grade the forms and the hyperlink doc where they're filling it out, or do you just grade the forms?
I graded both.
Okay.
So the Google form, the questions, there's one question, every question is one that can be self-grading on a Google form. I don't have them explained. Have I done that in the past? Yes. I don't want to grade it.
Mm-hmm.
So it is everything that they can just give me an answer for. The very last question is always, what was the best part of your lab today? And they write something. Only time I would grade that, I mean, they have to have something in there, is if I think I have a squirrely group, then I might go look at it, and if they said something stupid, which they can, then I'll dock them, right? But I don't hold myself to that. That's if I can just scroll down. You can just super scroll down through that. Usually at this point, they've been with me all year, and they know I mean business, and that I am holding them accountable for their benefit, and that we're all on the same team, and so just do what I ask you to do. So usually I have buy-in. It's very rare that it's ever a problem. So these Google Forms, you can grade every few days. I have a TA. I print it out to a spreadsheet, and I sort it by class period and by last name. So all you have to do is look to see who's in the group, and then write down their scores for me on a printout. And so all I do is then take that and I record it. So that part's pretty easy. This part I grade, and I gave you my key for this year, how I graded it. And I do not grade every single question. I sample. Okay, so there's no way I'm going to grade every question. Yes?
Do the forms ever inform reteaching or re-explaining for future labs, or are they kind of just a way to see that the student understood that specific lab?
They are more about that particular lab because each lab has its own Google form.
Okay.
And at the point that they have filled out that Google form, they haven't necessarily prepared for the next lab for me to be able to hold them accountable to it yet.
Okay.
Does that make sense?
Yeah.
Now, this hyperdoc, if you click on this or open it, I think we already have it. Okay, so this hyperdoc, you can see, right, they put their names here. It's collaborative. I talk about digital fingerprints. This is not the first time they've heard this because I have hyperdocs about plant hormones, or I have hyperdocs on DNA. I have hyperdocs on stress. So I use them intermittently. I mean, there's a whole thing we could talk about, just hyperdocs that I could talk about with you, but that's not it. We'll put a pin in that. But I just want you to know that they have heard all along digital fingerprints. And so I should be able to open up any hyperdoc and you look at version history, and I should see your digital fingerprint throughout. If I don't, and I see it 20% of the time, then once I have the grade, I will give you 20% of the team's points. It's very simple. It's really easy to do. Version history tells all. So if we go, can we go back up to the top? Okay, so this, you'll notice is prep work. So this was before day one. They watched the little Amoeba Sisters. This is a link to it. And then I had a series of questions. Let me tell you how I set up my hyperdocs to save me. Do you see the word type is in a different font and a different color? They have to click on the word type and type in that font, that color. If they change it, I won't grade it. Okay? And you teach that early on in the school year. And you're like, you changed it. Well, I was trying to be creative. I told you not to be creative here. There's lots of ways you can be creative. This is not one of them. Okay? I can tell exactly what they typed. And it's in a different font. It's easy for me to see on each of their answers on that hyperdoc. Okay? So there's a series of questions here, okay, then go right here. Day 1, some tools of the trade. This is also expected to be done before class starts. Now, I can talk to you about lab notebooking. I brought, where did I put them under here. Thank you. My kids, I use what's called lab maps and lab notebooking. And so this is how I do labs. And I have done it, and I have copies of how you can do it as a lab. If you don't know what this is, how many of you have seen these before? Okay. So I'm gonna let's just start passing these around. One of these has an Amgen. See, you guys are gonna pass back to there, and you guys are gonna pass back to there, okay? So this is just a blank one. What this is the students right here, and I make lab maps for every one of my labs, which you can have, okay, for any lab I do. And it's the flow chart of what they're gonna do in that lab. And they write here, and then below it, you see this yellow copy here? When they turn in, they rip out the yellow one. They keep, and this is what's done in college today, that's why I do it. They keep this white one here, and it has no writing on it. I grade this with a rubric. And before I start labs in a normal, I'll pass around, these are some former students' labs, so you can see. I have rubrics. So as far as photocopying goes, the only thing that I really photocopy is a rubric. And so at the start of any lab that I do, that's going like in their lab notebook, is they have to have everything done, the title, the background, the variables identified, their hypothesis, whether it's a null or a research hypothesis. They have to have their flow chart done, and I check their lab book. At that point, they have to leave them open and flat at their lab station, and here's the best part. I expect it to be done at the start of class. If you call me over when everyone in your team is ready. You see peer pressure? So I'm not checking half your team. When your entire team is ready. So let me tell you, they're on their whole team, especially that, slug. They will be like, you need to have your lab notebook ready to go at the start of class because she's not gonna, and then I literally put a sign up at the front of my desk and they put lab station 3 is up at the top. I walk to lab station 3, they all have their lab notebooks open, I put my initials go down through all the pages that I needed to check. And then I just go, who's next on the list? And then I just go through, and they cannot start collecting data until they have my initials on it. And so that is a really good way on lab day. I have lab maps, if that's what you want. I have enclosed in this program lab maps that I have done. So I'm telling you, I know how to do lab write-ups. I have found for Amgen and the complexity and what you have to do, that it was too much work in setting up the lab for each of the labs to put in a lab notebook. It wasn't worth the time and effort. I wanted them to focus, and so that's why I streamlined it to a collaborative hyperdoc. You will also see on here where it says lead lab. Let me tell you, this is a saver for you. The lead labs, if we're in a team of 3 right here, we're in a team of 3. She might be the lead lab. The lead lab, when they turn these in, they have to clip them together. Everybody in their team, like if this was a team, they have to clip them together. That's how they turn them into me. When I grade them as a team because they all write the same thing 1,000 times and then you've got to grade it, and that's annoying. So what I do is I take the lead lab and I grade the lead lab top to bottom, every single thing on the lead lab. Then what I do is I look at the other labs, I pick different things every time. Maybe it's a data table, maybe it's a, here's another one. Maybe it's, you'll see at the top if you look where it says lead lab and then over to the right, how many points. So the lead lab is contributing 15 of their 25 points. And then next time if she was the lead lab, that's all I record on a a sheet of paper, I put a check next to their name if they've been a lead lab. They cannot be the lead lab again until the next person is the lead lab in whatever team they're in. So it rotates the pressure, so you don't have that one that's super, super perfect and like has a seizure and starts breathing rapidly if they lose 1 point. Do you know that person? It might be you. Okay. So that way everybody gets, and let me tell you, that's peer pressure, which can be very effective to bringing up the quality. And if I can't read it, do you know the answer to that? If I can't read it, I
Don't grade it.
absolutely right. Do not waste my time. You know, do not waste my time. Okay, so any quick questions? Because I have a little bit more to show you on this. Yes?
So were you saying
Wait, let's listen in case this is your question. Let's listen.
were you saying for the Amgen labs you just rely on the
Hyperdoc.
hyperlinks and the docs and you don't do here?
I don't. I
Okay.
used to.
Yeah.
It's like sometimes when I talk, then people are like, lab notebook, you know, it's really important. I'm like, I know, okay? But you know what? You don't have to be perfect teacher every time. You don't. You have to have a life, right? You have to be able to grade where it's not killing you. And so for this particular lab, what's my goal? I want them to understand these lab processes. I want them to feel empowered. They feel so empowered when they do these labs that they can actually manipulate an organism. When we have God Day, you know, they are so excited. That's what I call it. It hits all realms, believers, non-believers, they're all okay with God Day. But they know they're changing an organism, right? They are so excited that they can do those skills. I have goosebumps right now. That they're talking about it with their family. And you know what I'm hoping? I'm hoping when they have to go to a voting box, or they're voting by mail, hopefully, and they're doing those votes, that they are voting with an educated mind, making good choices for the rest of us, right? So I want them to be literate. I want them to understand it. They're so excited when they go to the doctors and they know the equipment that's sitting there or something. They're with their dad or their grandpa at the doctor. They're like, yeah, I've done it. And they're like proud, you know? They're so proud. So yes, lab notebooking is important. It's an important skill set. For this particular lab, I'm hyperdocking, and they work on it collaboratively, and then I grade it at the end. Okay, so we can hit the lights again if you guys are good with that. We can go back to that hyperdoc if you wouldn't mind. Am I over time yet? I've got 1 minute. Yeah. So if you scroll through this, you will see, let's go to like any lab 2 or something. You will see where they have to put in pictures or okay, start right there. Image of loaded gel before you turn the mini one on. I want to see a picture before, and I want to see a picture after. They can use their Chromebooks, they can use their phones, I don't care. But they have to have a watermark, so that way they can't use somebody else's image from a previous year. So that watermark is in each one of their images. It's something unique. It can't be like a number 2 pencil, because everybody has one of those. Has to be, you know, an earring or something like that that they use in all of their pictures. And so any modifications, here's that what I was talking to you about, task 4. Identify what each member of your team did today to contribute to your hyperdoc, your lab, and your Google Form. Everyone should be contributing and working collaboratively. Do not divvy up the work. Write name and contribution. So they have to write their name and what they did. And I said everybody else in your team is going to look at it, so it better be authentic. And then this is a link to the Google Form that is the closure. And then once they're done with that day's lab and everything is okay, then they can start prepping for the next day's lab. This is a link to the lab book, the ABE lab book. So I put hard copies in because I happen to have hard copies. I put those at the lab station for those kids that want that, or they can do the digital link. This is what they have to do before they come to class that preps them for the lab. And then if we just kind of scroll down through it, you can just kind of see that this is on repeat. Keep going, you can keep going. Beautiful, lab 3. Here's gel electrophoresis day. Little images, how I want them to load it. They have to insert an image of their completed gel. Here's their analysis that I have to have them do, but then also here is a Google form that asks them specifically about those things. All right, so that is what they work on while they're doing the lab. And then if we go back to the presentation, please help me maintain my integrity here because this is like a key, so to speak. Yeah, I mean, for sure it is. So click on that. And this is just explaining where my points came from. So this was worth 50 points. So these are the things I graded and how many points they were worth. Okay, so if you keep just kind of scrolling through that a little bit so you can see. And then the way I grade in HyperDocs, this is how it helps me. I highlight the question. If there's a problem with it, I just put the problems. I highlight the question, I make a comment, and I might say -2. And I write why they lost those points. -1, incomplete. You know, -4, didn't do, or whatever it is. Then all I do is, then I just add up all the minuses and take it away from the plus. I usually have some embedded, like 10 points just for doing it, you know what I mean? And then this is how they are curating their lab. And you can keep going all the way through. You can go faster. So you can see how these are the kind of, I took this from a student paper. That's their watermark. So nobody else can use that. And then you can see I had them set the microbyte better. Okay, keep going. Keep going, you're good, you're good. Here's their gel. You can see watermark. Keep going. So there are different images. They had to put an image of Piara. They had to put in an image of Pecan, so they had it to use. We can keep going. So there's one student's gel electrophoresis. They had to label what they had in each tube. And we can keep going and keep going. So here we can pause here. They had to put their plates in here. So it's a picture of their plates. Early on, they had to do their predictions. And then before they, you know, the day they played it out, but they didn't know their results, they had to do their predictions. And then they had to do their results. I got to tell you, it's like one group out of 8 in all my classes that don't get the red columns. You know, they usually are pretty good at it. Okay, except remember when I told you I screwed it up. Right. I've done other screw ups too, but we just don't have time for those. Okay, let's keep moving. And then at the end, I have them do plain evidence reason about their lab. You know, were you able to make a recombinant DNA? What is your evidence? What is your reason? Okay, and then back in the presentation, so you have that right there. It says key. Okay. This right here explains my lab notebooking processes if you were interested in lab notebooking because we talked about it. So this explains how I do lab notebooking. This explains the 5 elements I like in flowcharts. So that's a document link that you can have here. This is a link to all the ABE lab maps when I did it. Originally I had a lab map for each one. And then I broke it into 2 lab maps where it was the first 4 labs and then the 5th lab. I don't do the protein purification because I just don't have time. I think it's lovely, I just don't have time for it. But if you click on this, that is a link to that. Okay, and I think there's one more page. Any questions, thoughts, concerns? Yes?
Can you go over how you can tell which students have worked on it more or less?
Oh, yeah. When you go into a Google Doc, here, go into any Google document. Okay, go to File, Version History, File, Version History, see Version History. See, Winnie Sloan did all the work on that document. So you can scroll down here, and you can even hit the down arrow on this one. You can see like individual edits of that one grouping. So I know who's contributed, and again, I'm just telling you, you only have to, this is, you know, you only have to nail somebody once, lesson learned, you know, because they see it, and I'm like, sorry, you know, I can't, you know, I'm real, like, not mean, you know, I can't give you points for something you didn't do. Well, I was talking, and then they wrote it down. I go, well, that was a poor choice, because I told you digital fingerprints. So next time you'll have, and then they will ask me, like other labs or other documents, they'll say, is there, I'll tell them, hey, on this one, like the fetal pig lab, I have them make a, they make a Google slideshow of their fetal pig dissection because I found that to be most effective and they have to label things on it. So they usually have one person in their team during fetal pig who's like, no, I cannot dissect a pig. Let me tighten my leather belt, you know? And so I have them, like if they do all the recording on something like that, then it's okay because they're not going to contribute to the cutting part. You know what I mean? So they have to get the okay. Otherwise, I check version history. Any other I know, it's time.
It is time.
Okay, any other questions or concerns before you go?
Thank you.
You are so welcome. Good job. Listen to me. Good job you for taking this on. I know it's a challenge. It will get easier over time. I believe in you. You're welcome. If you wouldn't mind